morning, and welcome back to LMU News Live. Tonight, we're kicking things off with an inside look on the body's own communication superhighway. Reporter Bella is standing by with a special report on how your nervous system keeps you alive, alert, and well, mostly sane. Bella, what's the latest? Good evening. Tonight, we bring you a special report on the two biggest communicators in the human body, the nervous system and the endocrine system. Though they use different methods, both are dedicated to one mission, keeping your body informed and in balance. Sources confirm the nervous system performs three crucial jobs: sensing the world, processing information, and ordering the body to act properly. This system is divided into the brain and spinal cord based off of the central nervous system and the nerve network known as the peripheral nervous system. The PNS has two roads: the sensory division, which brings information in, and the motor division, which sends commands out. And in the motor division, we even have a spicier rivalry, the somatic division, which controls voluntary movement, and the automatic division, which handles smooth muscle, cardiac muscles, and glands. The autonomic division itself is split into two feuding parties. The sympathetic division revs you up for fight or flight, while the parasympathetic division cools you down for the rest and digest. Together, they keep you alive in a world full of stress, snacks, and drama. Thank you, Bella. Now let's switch gears and head into a high-stake crime investigation inside your own body. Joseph, our reporter here, joins us from the Neural Crime Lab with breaking updates on the fastest communicator in the human body. Joseph, what are you uncovering today?
After hours and hours of CSI Neuron Unit's research, we're live inside this Neural Crime Lab, inspecting the mysterious suspect behind every thought, feeling, and reflex, the neuron. The cell has three major abilities. It can respond to stimuli, it can conduct electrical signals, and it can secrete neurotransmitters to pass messages along. Our lineup of suspects includes sensory neurons, seen here, that deliver incoming information, interneurons that serve as the brain's analysts, and motor neurons that deliver outgoing orders. Under close examination, the classic multipolar neuron, as in here, includes dendrites, the receivers, a soma, the processor, an axon, the messenger wire, and axon terminals, the transmitters. Other structural suspects are unipolar and bipolar neurons. Each play unique roles in neural communication. Meanwhile, a team of neuroglia is on the scene, offering full-time support. In the CNS, we have astrocytes, oligodendrocytes, edemal cells, and microglia. In the PNS, Schwann cells are seen here, and satellite cells take charge. Schwann cells can also handle nerve regeneration, a privilege that the CNS unfortunately does not have. And good news, if the axon is myelinated and wide in diameter, the messages travel with lightning speed. Case cracked. That's all I have from the CSI Neuron unit. Back to you guys for the rest of your segments. Thank you.
Fascinating work, Joseph. For our next segment, we turn to the brain's electrical engineering department. Signals, voltage, and split-second reactions. How does the brain keep the power running? Reporter Gil has the electrifying details live from the tech desk. Gil, what can you tell us today?
Tonight's report dives into the world's most advanced electrical grid, the nervous system. Every neuron maintains a resting voltage gate of about -70 millivolts. This is also known as the resting memory potential. This delicate balance is maintained through sodium and potassium ion gradients. You have potassium leaving the ICF, and you have sodium coming into the ICF at the same time. When a stimulus hits, a neuron generates a local potential, a tiny electrical change that may or may not spark something bigger. That's known as hitting the threshold. So say the stimulus does hit the threshold. This is going to release action potential, and the sodium channels are going to open, and this is going to cause a rapid depolarization. Once it hits around 30 millivolts positive, the sodium channels are going to close and the potassium channels are going to open. And this is going to cause a rapid repolarization. So the cell is going to become more negative as the sodium channel or the potassium channels open. This is going to dip past resting membrane potential, which is going to be called hyperpolarization, and then it's going to go up back to resting membrane potential. And it's kind of an all or nothing system. So if the local potential fires but does not reach threshold, the stimulus will die and it'll reset, and the action potential will not be stimulated. Okay, we also have a discussion about continuous versus saltatory conduction. Basically, this is just referring to the unmyelinated axons that conduct signals continuously. So continuous equals unmyelinated. It's a fuse burning down a rope. Myelinated axons, however, use saltatory connections, where the signal leaps from node to node like a superhero on a rooftop. It's much faster. And thanks to the refractory period, signals can only move forward, preventing message backtracking. So you can see the difference in impulse between continuous with the unmyelinated and the myelinated. The myelinated is a lot quicker and more often.
Thank you, Gil. Now we're zooming into one of the busiest hotspots in the body, the synapse. Millions of chemical messages fly across in every second. Reporter Luca is live in the scene to show us how these microscopic press conferences shape everything we do. Luca, what's happening at the synapse tonight?
We're reporting live from a packed synapse, the microscopic space where neurons hold their biggest press release. The presynaptic neuron arrives with the neurotransmitter-filled vesicles, releasing chemical messages into the synaptic cleft. Popular molecules at the scene include acetylcholine, glutamate, GABA, serotonin, dopamine, and several neuropeptides. Each neurotransmitter's effect depends on its receptor, meaning the same chemical can either excite or inhibit depending on where it lands. At excitatory synapses, neurotransmitters cause depolarization and create EPSPs, bringing the neuron closer to firing. At inhibitory synapses, neurotransmitters like GABA create IPSPs by hyperpolarizing the cell. Some synapses use second messenger system to amplify the message, and the transmission ends through diffusion, reuptake, or enzymes. Neurotransmitters such as endorphins move through in the background, adjusting mood, pain, sensitivity, and overall synaptic strength.
Incredible insight, Luca. For our final segment tonight, we explore how all these signals turn into memories, decisions, and the very core of who we are. Reporter Martha brings us in-depth look at the neural integration and the grand finale of the brain communication. Martha, take it away.
Hi everyone, I'm Martha. I'm thrilled to be making my AandP Dispatch debut today as I report live with breaking new news from scientists regarding neural firing and synapses. So let's get into it. First, we have to define what an EPSP and an IPSP are. Now, these two are antagonists. An EPSP is an excitatory postsynaptic potential, which you can see right here in red. It moves the postsynaptic cell closer towards reaching threshold, while an IPSP does the opposite. So an IPSP is an inhibitory postsynaptic potential, which cancels out the effect of the EPSP, bringing the postsynaptic cell further away from reaching threshold where an action potential can occur. So seen in this drawing here, an EPSP coming here into the trigger zone and an IPSP coming here into the trigger zone cancel out, and therefore no action potential results. All right, so when does an action potential actually occur? Now, a single EPSP will likely not reach threshold alone. It is only by stacking various EPSPs that we can reach threshold, and thus an action potential can occur. Now, there's a few different ways that these EPSPs can be stacked, though. The first right here is spatial summation, and this is when signals from multiple sources arrive at the trigger zone and together reach threshold, leading to an action potential. So here you have EPSPs coming from here, EPSPs coming from here, where they combine at the trigger zone. That's where threshold is reached. You can see EPSP from this source, EPSP from this source, action potential follows. However, there's another way that neurons can combine signals by stacking rapid impulses from a singular source. So that's right here. There's only one source here sending EPSPs right here, but by more rapid sending of EPSPs a singular source can also reach an action potential like that in temporal summation. All right, now let's talk memory because it's these synapses and processes that lay the foundation for one's memories. So synapses form memory by strengthening existing neural connections or by creating new ones, and that process is called synaptic plasticity. Additionally, repeated stimulation can strengthen these connections, and that's called LTP, or long-term potentiation, and that can cause neural changes, new connections, new receptors, things like that, that are eventually what make memory permanent. And let's talk about permanence because memory appears in stages, as we all know. So immediate memory are those things that last only a few seconds. Short-term memory lasts minutes to hours, and long-term memory is what can last a lifetime. And long-term memory consists of both explicit memories, those are facts you know and things like that, while implicit memory is also a part of long-term memory, and those are skills and things you've learned that you can just perform automatically. You don't need to think about it. It's second nature to you. And then forgetting obviously occurs, and that is scientifically when synapses either weaken or retrieval pathways from your memory, they fail, basically, or give out. And so it's a very complex network, but this memory, these synapses are what allow you to store every moment and memory and decision in your life. So it's really important that we understand them and we keep them in the back of our mind.
Thanks to all reporters for breaking down the incredible world of the nervous system tonight. From neuron investigations to electrical signals, synapses, and the circuits that shape our memories. It's a reminder of just how busy and brilliant our bodies really are. That concludes our special report on Chapter 12 Nervous Tissue for LMU News Lives. I'm Bella. Thanks for joining us and have a great night.